first want to start off with a statistic. Over 50 million individuals suffer from neurodegenerative diseases worldwide. These diseases are the loss of neurons in different regions of the body. And examples include ALS, Alzheimer's, and Parkinson's disease. In the context of Parkinson's disease, there is a specific loss of dopaminergic neurons in the midbrain. And this causes the motor symptoms associated with Parkinson's. Unfortunately, there currently exists no cures. However, with the discovery of induced pluripotent stem cells, researchers have managed to develop specific protocols to produce certain cell types, like neurons. And BitBio, our collaborative partners, have found a way to genetically modify these iPSCs to specifically become a specific cell identity, again, in the context of neurons. And then what you could do is create these neurons, insert them into a person who's got loss of, for example, in Parkinson's, loss of these dopamine neurons, and potentially treat the patient. And this is all great, but what are the issues? Unfortunately, most iPSCs screen primarily for genetic abnormalities in the nuclear DNA. But what about the mitochondria, these little batteries in our cells? The mitochondria contains DNA, and each mitochondria has its own DNA. And this mitochondrial DNA accumulates 100 times more mutations than the nuclear DNA. And as there are multiple mitochondria, different amounts of mutations accumulate in each mitochondria individually, causing a heteroplasmic amount of mutations in the cells. My project's investigating how this heteroplasmy in these iPSCs might affect the differentiation potential of the different cell types. And the way we want to look at this is causing a mutation in the mitochondrial replicative machinery in these iPSCs, which is inducible. So when you add this drug, doxycycline, these cells will then have accumulation of mitochondrial mutations. And we will then produce a method of reliably assessing how the mitochondrial mutations affect this differentiation, as well as sequencing this DNA and finding a quality control measure, which can then feed back towards this way of presenting cell therapies for different approaches. And that is the point of my project. Thank you for listening.